So one thing I wanted to do was compare two types of cells, the ones that we've been talking about most often, the pacemaker cells, and right next to them the myocytes. And you're going to start seeing some really interesting kind of similarities and maybe some differences as well between the two. So let me actually, on this side, we'll do our kind of usual millivolt scale, positive and negative, right? And we'll do the same thing over here. And on the left, we're going to do our pacemaker cell, and on the right, we'll do our myocyte. So let's do our pacemaker cell first. And you remember the pacemaker cell kind of starts out somewhere around -60 and kind of creeps up, right? We've drawn this a few times now. And then it hits its action potential, and it kind of goes up more rapidly at that point, and then finally starts coming back down again. And this is kind of the pattern that repeats over and over. And on the other side, we've got our myocyte, our muscle cell. And this is kind of the cell that does a lot of the heavy lifting, as it were, for the heart, kind of what you imagine the heart cell might be. So this myocyte actually starts out a little bit more negative, around -90, let's say, kind of is more flat initially, right? And then it hits its action potential, and it rises much more steeply than the pacemaker cell. So now that they're kind of side by side, you can see the difference, right? And then it comes back down as the potassium begins to leak in. Except there's a new phase here because the potassium and calcium kind of offset each other, and they begin to kind of have this plateau. And eventually the potassium kind of wins out, and then you get repolarization where it basically gets back down to where it started. So this is kind of a very rough schematic of what it would look like. And let me actually now bring some space onto our canvas. I'm going to make some space here. Let's actually draw the two cells. So we've got a pacemaker cell over here, and I'll draw the myocyte right next to it, right? And this is our myocyte. And I'm drawing them the same size, but that's just so that you can see things clearly. So what's the first thing that happens with our pacemaker cell? Well, you know that when it's rising, when that membrane potential is rising, sodium is actually coming in, right? And I'm going to draw a little set of lines here to represent sodium coming in. So sodium is coming in here. And then you get a rapid rise, right? You get that rapid action potential. And for that, I'm actually going to draw white lines to represent the voltage-gated channels. So this is kind of my way of representing voltage-gated channels. And here you have predominantly calcium coming in, right? And I'm going to take a quick moment to pause here and say, when I say sodium is coming in or calcium is coming in, I don't want you to think that that's the only ion that comes in at that point. I mean, when I say sodium is coming in, for example, that's the main ion that the cell is permeable to, but it's not the only one. In fact, we've even talked about the fact that when sodium is coming in phase 4, that sometimes a few other ions are actually kind of leaking in and out as well. So just keep that in mind. When I kind of draw one ion, it's not to say that that's the only one. It's just to kind of make things a little bit more clean and clear so that you can kind of get a sense for what's the overall gist of what's happening. So back to our kind of regularly scheduled program. We have now another voltage-gated channel down here. And this is our potassium that's leaving, right? So this potassium leaves. And to make it really kind of clear and parallel, let me actually go one step further and draw on here the phases. So we have phase 4 here, and this is phase 0, and this is phase 1, right? These are the phases of our action potential. And phase 4, the main ion we said is sodium. Phase 0, the main ion is calcium. And phase 1, the main ion is potassium. Now, a cardiac myocyte initially has mostly potassium leaving, right? So the dominant ion here is going to be potassium leaving. And that's what's setting that membrane potential. And then you have in the action potential, actually let me switch colors to a voltage-gated white channel. So these are the voltage-gated channels now. You have sodium entering the cell. So here sodium is entering the cell, like that. And then you have in phase 1, you have now some voltage-gated potassium channel. So unlike the one at the top of this cell, now you've got potassium leaving just as before, but these are voltage-gated. So they kind of flip open and flip closed based on voltage. And then you finally have another ion over here coming in, which is calcium. So you have calcium coming in as well. And so let's do the same kind of exercise we did before, where you kind of go through and label the phases. So we know we have phase 4 down here, phase 0 here, 1, 2, and 3, right? So this is 1, 2, and 3. And what would this potassium be? Well, this potassium is the dominant ion in phase 4. And sodium is happening, or sodium coming in is happening during that action potential, during that phase 0. And then potassium, these voltage-gated channels, they're kind of involved in phase 1, 2, and 3, right? That's kind of a unique property of those channels. And they're not the exact same channel family. They're different families of channels. But their voltage-gated potassium channels are actually kind of involved in a few different phases. And this calcium is involved in phase 2. So now you can see kind of how the different channels are involved and also their action potentials kind of side by side. Now, one thing I should also kind of point out is that in the myocyte, and this is less true of the pacemaker cells, and I say less true because they also have this, but it's unclear what the role is. They have this thing called a sarcoplasmic reticulum. Sarcoplasmic reticulum. And a sarcoplasmic reticulum, I think of as a magnifier. Sarcoplasmic reticulum. Sometimes you'll see it as just SR, reticulum. A magnifier. Well, what do I mean exactly? Well, what happens is that this sarcoplasmic reticulum, I think of it, it's an organelle basically. It's sitting inside of a cell. It's an organelle. And this sarcoplasmic reticulum is a bag of calcium. Literally is a bag of calcium. So this is sitting here with all these little calcium ions inside. And what it's waiting for is a signal from the cell to say that some calcium has entered. So once this calcium has entered, what it does is it binds to a little receptor, binds to a receptor right here. And when it binds to the receptor, calcium from the inside of the SR, or sarcoplasmic reticulum, is dumped out into the cell. So why is that necessary? If you have calcium already coming in from the outside, why would you need more calcium coming from this sarcoplasmic reticulum, this bag of calcium? Well, what happens is that this bag of calcium can basically empty out really quickly. So you can have just a few ions kind of trickle in from the outside. As long as they bind to that sarcoplasmic reticulum and let it dump out, then you get tons of calcium kind of flooding into the cell. So it basically magnifies the effect of calcium, right? So just keep that in mind, is that when we talk about phase 2 and this calcium entering, the one thing that I haven't really talked to you about until right now is that there's this magnification that happens because of the sarcoplasmic reticulum. All right, now let's bring up kind of the main reason I wanted to do this video, which is when you have all these ions kind of floating in and out, you may be wondering, well, how in the world does the cell actually reset itself? I mean, at some point, doesn't it need to kind of get things back to the way they were? Otherwise, you'll just run out of sodium and calcium on the outside, and you'll just fill up the cell with that stuff. In other words, if sodium and calcium just keep coming in and potassium keeps leaving in this pacemaker cell, at some point, won't you have no gradient left? And so how do you set up those gradients? I mean, that's the real question, right? So you'll remember that there are these little ATP pumps, and a pink pump is basically going to be, for me, my code for using energy. And it's going to be throwing out sodium. It's going to be throwing out 3 sodiums and bringing in 2 potassiums. And there's also a little pump over here, that does something very similar, but for calcium. And what it does is basically kind of just boots out calcium, says, see you later, buddy. So this calcium leaves over here. And actually, there's another strategy, there's another pump right here. I'm going to draw it right here, that also gets rid of calcium. But this one is not pink. This one does not take energy. So you're thinking, well, how in the world did the first 2 take ATP? How do these 2 take ATP and this third one not? Well, you can think about the fact that there is a sodium gradient. Sodium likes to get inside the cell. We know this, right? And it likes to be inside the cell because you've set up a sodium gradient because of this right here. So if you've created a sodium gradient using energy, you can also use that sodium gradient to drive out calcium. So you have a couple of mechanisms to take care of our ion problem. So for example, we needed to figure out how to get rid of this sodium, and we've got our answer right there. We had to get potassium back into the cell, and we did that right there. And we also had to get rid of all this calcium that keeps coming in, and we did that right there and there. So this is how our cell takes care of those ions. Now what about our myocyte? How does that work out? So how does it kind of reset all the concentration gradients after all this stuff happens as I described above? Well, it also, of course, has, our two ATP using pumps, right? So these two are using ATP, and it's going to also drive sodium out. And let me just draw that right there. So 3 sodiums leave. This is all looking kind of the same, right? 2 potassiums enter. And already I've kind of solved some of my problems. And it has, just as before, it has this kind of sodium-calcium pump. So calcium exits right there, and sodium enters right there. So kind of the same answer as above, but as to the left, but you also have this, remember this sarcoplasmic reticulum, right? And this sarcoplasmic reticulum, I said, was loaded with calcium. Loaded with calcium. Well, how did it get loaded with calcium? How did that even happen? Well, the way that it happens is that there's actually a pump here. A pump, let's draw it right here on this side, that basically pumps, using energy, pumps calcium inside. So you can actually pump calcium into the sarcoplasmic reticulum using another ATPase. So it's very similar to the one that's on the membrane. They look literally the same, right? Except this one is on the sarcoplasmic reticulum. And doing our same kind of checklist, you can see that, look, sodium is pumped out, and that takes care of this guy. And then we have to bring potassium back in, and that's done there. And then we have to get rid of all this calcium, right? All this calcium that kind of came in here and here. How do we take care of it? Well, we pump it out there, we pump it back into the sarcoplasmic reticulum there, and we can exchange it for sodium there. So this is how we kind of take care of and literally reset our cells. Now, the final thing I wanted to say is that if you actually think about it, if we're talking about permeability, let's say we want to talk about whether more calcium is coming into a cell or out of a cell. Well, usually, under most circumstances, we think of these guys, all these kinds of energy-driven processes or using concentration-driven processes, kind of operating at a certain rate, right? They're kind of always operating at a certain rate. And the same is true for these guys, right? We only have one extra thing over here, but they're kind of operating at a certain rate. Now, if I increase the permeability, let's talk about calcium, for example, of calcium. Let's say increase the permeability of calcium. That means more calcium is kind of entering that space. And if I decrease the permeability of calcium, I'm talking about this channel right here. If I decrease the permeability of calcium, that means less calcium is entering that space. Now, would you accept the following? What if I told you that I'm not even going to change? Let me erase that little line I drew. I'm not even going to change how this permeability is going to function. All I'm going to do is, what if I change this? This is kind of an interesting idea. What if I change how fast this guy works? How fast he works? Well, if he starts working more sluggishly, let's say he's working more sluggishly, then you have more calcium kind of hanging out over here, right? You have more calcium hanging out in the cell. And let's say you make him work really fast, really fast, so he's pumping that stuff back in. Well, then you have even less calcium. Now you have the opposite. You'd have less calcium kind of out here. And so you can actually now see how by changing the basal rate of these pumps, you can also affect the amount of ion that's hanging out in the cell. And that, of course, is going to affect the membrane potential of that cell. So just keep that in mind, is that usually we think about this. We're usually thinking and talking in terms of this stuff up here. What is the permeability? What ions are coming in and out? And we kind of assume that this is static, that this stuff is not changing too much. But every once in a while, you'll see that resetting the membrane or resetting where the ions should be, you can actually tweak those mechanisms as well. You can actually make some of these pumps work harder or less hard. and that's going to have an effect on the amount of ions as well.